everyone. I'm Gbolaga from Ohio University. I'm a space biologist, a bit different from astrobiologists in that we send plant samples to space and we try to see how those plant samples adapt to the spaceflight environment. And then we use information from these to make them better adapt to extreme environment on Earth, or to also prepare how we can cultivate plants in space for future cultivation, future exploration. And so I work on Arabidopsis plants, which is a model plant. It's what E. coli is to microbiologists. And it's model because it has all the genome sequenced already. And so we can easily trace a particular phenotype to a particular genotype. So to the right up there is what the Arabidopsis seedlings look like after we get them back from the space environment. In this case, I mean about the International Space Station at the lower Earth orbit, where we have zero gravity, microgravity, and where we have ionizing radiation and altered flue gas dynamics as one of the extremities in that environment. So basically, we send seed to space. We let those seeds germinate in space. We let them grow for a few days. We freeze them, and we return them to Earth. And when they get back to Earth, I analyze them, transcriptomics and proteomics, to make sense of how such plant adapts to the spaceflight environment. And for us to conduct spaceflight environment, we use different hardwares, as you can see. On top here is the Advanced Plant Habitat. That's the most desirable hardware currently for almost everyone researching space plant biology. And here we have the brick hardware. I love using the brick. It got the name from the fact that it looks like a brick block. It's biological research in canister. I work with it. And this is the European modular cultivation system. It's being retired currently and being replaced by the multivariable platform. So these are just few of the many hardwares that we use to send specimen to space, plant, micro, but in this case, we use this to send our plant to space. The first multi-omics experiment conducted on plants sent to space was conducted by my lab, and then we discovered that comparing the log4 change of the transcript, that's the RNA, to the log4 change of the protein, while plotting a correlation graph of it, we discovered very low correlation between differentially expressed transcript and differentially abundant protein, which is indicative of the fact that post-transcriptional modification of transcript is one of the adaptive mechanisms of plants to spaceflight environment. That means not all RNA gets to become protein. And that also, it's underscore the need for us to study proteomics, to conduct proteomic studies of these plants that we send to space, because the proteomics will probably give more insight into the adaptive mechanism than the transcriptomics. Unfortunately, because proteomic study require large tissue mass, which we don't have the luxury of having large plants sent to space, most people conduct just transcriptomic study. Until date, globally, only 4 proteomic studies have been conducted on Arabidopsis aboard ISS. And these are the 4 proteomic studies. Different environment, different conditions, different variables, age of the plant, hardware, light condition, seed line used, the tissue type, and the type of protein extracted. They vary across all those 4 experiments. 2 of those experiments were conducted in the lab. I'm the one in charge of the last one in red, called the Brick LED project. So to get idea of to get idea of plants' response on the proteomic level to the spaceflight environment based on these field data set, we have to do what we call meta-analysis of the proteomic data set from these from these 4 experiments, and that's what I do. So this is the first stage of the meta analysis. So we start from the left here. We intersected the protein abundance with the detected protein across all the 4 hardwares. And common to the protein across all the 4 hardwares these are 3 proteins, which are PCAP1, that's the microtubule destabilizing protein, which is involved in chromosome separation during cell division. It's involved in formation of cell shape, and it's also involved in transport, vesicular transport in plant. And also we discovered that common to these 4 data sets, protein, is that the protein involved in defense response and mRNA binding. Now, I went a step further and isolated the data set called EMCS, which is the data set called ABRS, Advanced Biological Research System. I isolated it from the general data set because they used a mutant seed line. So I compared those seed lines that were the wide type, and from intersection of the 3 data sets that use our seed lines that are wide type, we have these 8 proteins, and here are the proteins. So conducting a protein interaction network analysis, we see that regulation of microtubule destabilization, microtubule rearrangement is a major adaptation mechanism of plants to the extreme spaceflight environment. And one thing we notice in phenotype of plants that we grow in space is that they are usually smaller, looking less healthy compared to those grown on Earth, where we have the full gravity and no ionizing radiation. Now, I went further to compare the brick data set, which are the one that we conducted in our lab and we can be so sure of. So we have 2 of them. We have the brick LED, which were conducted in light. You know, this brick LED hardware had light in it. And we have the brick PDFU, which had no light. So all these plants, these experiments were both flown to space, flown to space, but one had light, one had no light. Both of them had no gravity. At the intersect of them, the protein here, 186 of them, will definitely be due to the spaceflight environment. While the protein here will be due to photomorphogenesis, that is growth in light, cross-interacting protein, maybe between the hardware and growth in light, and elusive protein due to the advanced technology that we use in the brick LED compared to the brick PDFU. While the protein here are those involved in scotomorphogenesis, growth in darkness, and cross-interacting protein. Now, I call this protein, due to spaceflight environment, I call them the brick intercept. So, comparing the brick intercept, this is the brick intercept, comparing it with another data set from the European Modular Cultivation System, where they had a centrifuge aboard the International Space Station, 1G centrifuge. Having a 1G centrifuge above International Space Station eliminates microgravity. So it introduced 1G gravity. So this data set, EMCS, has excluded microgravity. And everything in this data set is radiation. So comparing, intersecting this brick intercept with this EMCS data set, whatever is in the middle here will definitely be due to radiation or altered fluid gas dynamic. You know, in the absence of gravity, plants don't pick water the same way they will do on Earth. And whatever we have here is due to microgravity, definitely. And whatever we have here, proteins here, are due to the EMCS hardware and cross interaction. Now, doing a protein-protein interaction network of the proteins that we believe are due to radiation, we see that most of them are involved in ribosome assembly, ribosome biogenesis, and protein translational machinery. So one takeaway from this is that Arabidopsis ribosomal activity is altered by spaceflight ionizing radiation. So currently, I'm comparing the transcriptomic data between these 2 data sets, and we're also trying to do a wet lab determination of the specific role of this intersecting protein. So we're hoping to collaboration on a proteomic meta-analysis of this spaceflight stuff. There are a lot of things that could be done on it. So my acknowledgement to Dr. Sarah White, that's my advisor, and then Dr. Colin Cruz from the National Lab in New Mexico, and to the NASA Analysis Working Group, Plant Omics, who sponsored this research. Thank you so much.